Well, hello. I'm Dr. David Ellison, professor of medicine and chemical physiology and biochemistry at Oregon Health and Science University in Portland. I'm also a deputy editor of the Journal of the American Society of Nephrology. And today I'm joined by Dr. Assam El-Osta to discuss his exciting paper entitled Set7 Methyltransferase and Phenotypic Switch in Diabetic Glomerular Endothelial Cells, which appeared recently in JASN. Dr. El-Osta, why don't you introduce yourself and tell us a bit about your background?
Good morning, David. It's a real pleasure to join you in my inaugural podcast with JASN. I'm a professor of human epigenetics here at Baker Heart and Diabetes Institute. I lead the program in understanding the role of epigenetics and particularly chromatin biology and how this influences transcriptional control in human disease. And a focus for the laboratory is trying to understand the role of epigenetics in diabetic complications.
Great. Well, your paper does focus on the role of a protein I wasn't familiar with, set domains containing 7 histone lysine methyltransferase, in contributing to diabetic kidney disease, which is the most common cause of end-stage kidney disease in humans. And while most of us are familiar with diabetic kidney lesions affecting the glomerulus and glomerular endothelial cells, you focused on Set7 methyltransferase protein. Could you provide some background about why this protein was chosen for study and how its activity is altered in patients with diabetes mellitus?
Sure. Vascular complications of diabetes are the most significant cause of human morbidity and mortality, affecting multiple organs, persisting despite tight glucose control. And that's possibly related to prior episodes of hyperglycemia. Now, this has been interpreted by many, including our team, as a memory of past glycemic control, known as metabolic or hyperglycemic memory, which is a major determinant of subsequent vascular events, and this involves the kidney as well. Now, epigenetic modifications control gene activity in metabolic disease, and our team has identified histone H3 lysine 4 methylation by the Set7 methyltransferase as a key regulatory determinant that is associated with transient hyperglycemia and the induction of persisting activating epigenetic changes of pro-inflammatory genes in human and animal models. Now, our studies have shown the signal pathways influenced by hyperglycemia are actually mediated in part by epigenetic programming. That activates the pathogenetic pathways involved in diabetic vascular complications. And the Set7 lysine methyltransferase is a metabolite-sensitive hyperglycemic sensor that is associated with pro-inflammatory gene expression. And our studies have shown high glucose conditions mediate Set7 signaling by writing the methylation marks on histone H3 lysine 4. Now, Set7 is also known to activate key pathways that many of us would be familiar with, including NF-kappa B, as well as the expression of pro-inflammatory markers and oxidative stress pathways in people with atherosclerotic vascular disease and type 2 diabetes mellitus. The effectiveness of Set7 to regulate these pro-fibrotic pathways relies on its methyltransferase capacity to tightly control lysine methylation on histone and non-histone proteins. So finally, given its contribution to pro-inflammatory gene activation in response to hyperglycemia, we believe Set7 is a potential target to reduce the burden of diabetic kidney disease. However, how Set7 controls pathways in the kidney remains poorly defined. And our study published in JASN, we've shown using single-cell transcriptomics, Set7 regulation in the mouse model of diabetic kidney disease, including preclinical validation by a pharmacological inhibitor and using a genetic knocker of the Set7 lysine methyltransferase.
Well, you've already answered one of my later questions, which was about the episodic nature of the hyperglycemia and whether that causes sustained effects. We'll come back to that lae on. For some of these studies, you used mice in which Set7 was deleted, knockout mice, and you crossed these with ApoE knockout mice. And then you used streptozotocin to generate diabetes in the double knockout mice. So I thought you might comment first on what's the physiological role of ApoE and why does its deletion accelerate the development of diabetic kidney disease, and why did you need to use the double knockout mice here?
Sure. ApoE is or apolipoprotein is a protein involved in lipid metabolism and transport in the body. And ApoE plays a central role in regulating the metabolism of cholesterol and other lipids, including their transport and redistribution among tissues. Now, in diabetic kidney disease, there are dramatic changes in renal lipid metabolism and transport. And this is why we focused on ApoE expression, which is protective against kidney damage in the context of diabetes. In mouse models, the absence of ApoE disrupts physiological lipid metabolism and transport in the kidney. And that disruption also activates pathways associated with inflammation, oxidative stress, and the accumulation of lipids. Now, these processes contribute to the acceleration of kidney damage and the progression of diabetic kidney disease in mouse models. Furthermore, ApoE deletion may also impair repair mechanisms and therefore intensify the progression of diabetic kidney disease. So overall, the absence of ApoE accelerates the development of diabetic kidney disease in these mice because of its role in maintaining physiological lipid metabolism and protecting against kidney damage.
So in your first figure, figure 1, you show that deletion of Set7 leads to a very striking protection from diabetes-induced kidney dysfunction. So that talks about what happens when Set7 is deleted, but what do you think the physiological role of Set7 is? If deleting it makes the kidney disease better, why do you think we produce, or the mice produce, Set7 in the first place?
Yeah, that's a very interesting question. We know that Set7 is a hyperglycemic sensor that shapes the diabetic epigenome. So essentially a key regulator of the transcriptome. This shapes the epigenome by enzymatically writing to histone as well as non-histone protein methylation. So it's linking transcriptional control with the metabolism and in particular inflammation and fibrosis in pathology. Now, previous studies in vascular endothelial cells, for example, show that H3 lysine 4 methylation written by Set7 methyltransferase is actually mobilized from the cytosol to enter into the nucleus in response to episodes of hyperglycemia. And so we've hypothesized that under physiological conditions, Set7 is likely to be poised to activate methylation marks to regulate glucose homeostasis. And in that context, Set7 has been implicated in the regulation of key regulatory marks such as insulin sensitivity and glucose metabolism through its interactions with histone and non-histone target proteins. Now, in our most recent published article in JASN, we show glomerular endothelial cells express the highest physiological levels of Set7, and this corresponds with what we refer to now as an endothelial to mesenchymal transition, or this phenotypic switch, which is implicated in microalbuminuria generation in diabetic kidney disease. Our studies also show that the mesenchymal gene markers upregulated by diabetes in mice can be attenuated in human glomerular endothelial cells by using small molecule Set7 inhibitor called PFI2. So overall, Set7, we believe Set7 contributes to the maintenance of glucose homeostasis by regulating insulin sensitivity, glucose metabolism, and ultimately the expressions involved in glucose utilization and production.
Great. Next, your data also suggests that Set7 activates insulin-like binding protein 5 in glomerular cells, which leads to inflammation and glomerular damage. So it seems like you believe that IGF-BP5 is also a key component of this endothelial to mesenchymal switch that occurs during diabetes. Does this mean that the scarring that's observed in this disease is generated by endogenous cells that have transformed and is not responding to external fibroblasts that enter the glomerulus in the kidney?
Yeah, so this doesn't rule out renal fibrosis because we know that Set7 influences fibroblast scarring in the kidney, a common physiological process which is characterized by the excessive accumulation of extracellular matrix proteins. Furthermore, we also know that IGF-BP5 plays a role in regulating fibroblast activity and fibrosis in the kidney through its interactions with IGF signaling, TGF-beta, and ECM remodeling. Now, in reference to the earlier question, in preclinical animal models, Set7 tightly controls the glomerular endothelial transcriptome in the diabetic kidney. Firstly, we observed elevated Set7 mRNA expression in diabetic podocytes, as well as mesenchymal and glomerular endothelial cell populations using single-cell RNA sequencing. Now, in contrast, Set7 was barely detectable in proximal tubular cells. Now, despite this increase in glomerular endothelial cell numbers, Set7 knockout reduced the expression of genes that define endothelial identity in the diabetic animals. And in fact, we observed a separation of this identity, specifically glomerular endothelial cells, from the mesenchymal population with reduced gene expression in diabetic mice with a genetic deletion of the Set7 methyltransferase enzyme. This influence on the transcriptome was consistent with a phenotypic switch involving the glomerular endothelial cell population. So using the single-cell RNA-seq methodology, we identified two interesting glomerular endothelial subpopulations based on high and low IGF-BP5 expression levels. The expression of renal IGF-BP5 is central because this transcription factor regulates endothelial identity. And we showed that IGF expression was elevated in diabetic mice, whereas IGF-BP5 levels remain unchanged in the kidney cortex in Set7 knockout mice. We also show IGF-BP5 was predominantly expressed in the glomerular endothelial cell. Now, this is where it becomes actually very interesting for us because high IGF-BP5 levels account for more than 75% of the endothelial population in diabetic mice, and this was significantly reduced in the diabetic mice with a genetic deletion of the Set7 enzyme. Now, these observations were in striking contrast to glomerular endothelial population with low IGF-BP5 levels, which was elevated in the kidney cortex of diabetic Set7 knockout mice. Again, using single-cell RNA sequencing studies, we confirmed the endothelial identity in IGF-BP5 high and IGF-BP5 low cell clusters. And we showed that by defining these high and low cell clusters, identify pathways that are activated for mesenchymal transition and other typical features such as reactive oxygen species generation, which were upregulated in the high IGF-BP5 endothelial cells, but downregulated in low IGF-BP5 cell populations.
So along those lines, and this is again reflective of the fact that this is not an area I study in my own laboratory, but I know that the phenomenon of epithelial to mesenchymal transformation generates controversy at times. What about endothelial to mesenchymal transformation? Is that a well-known and well-described phenomenon outside of this context?
In fact, this is a new description. It has been described in other contexts in the diabetic condition. So there are studies that have shown this endothelial to mesenchymal transition using technologies now that can actually delineate these signatures by using single cell sequencing technologies. So this is, I think, going to be an emerging area of future study for people interested in investigating diabetic vascular complications. And this has been shown in aortic endothelial transition as well as renal studies as well.
So you show provocative data, not just with Set7 knockdown, but also with a pharmacological inhibitor of Set7. You mentioned in the paper that the drug is not in clinical use. Can you provide any information about the drug, about its toxicity, about whether you see Set7 inhibition as a good target for drug development, and what potential limitations are present in the currently existing products that you used to inhibit Set7?
Sure, you're right. PFI2 is a small molecule inhibitor that targets the activity of the Set7 methyltransferase enzyme. And data on the toxicity of PFI2 vary depending on the context of its use in experimental models. There are few reports on its use in diabetic kidney disease, and in general, the drug toxicity needs to be addressed and assessed in preclinical studies before considering its potential for clinical development. Our in vitro and ex vivo studies show there is no cytotoxicity in endothelial cells stimulated with PFI2, and the drug concentrations that we've used in studies are ranging between 10 to 100 micromole of PFI2. With respect to Set7 inhibition as a target for future drug development for diabetic kidney disease, the small molecule inhibitor has not been approved by the US FDA or any other regulatory agency that I'm aware of for clinical use. In fact, PFI2 is primarily a research tool used in laboratory studies to investigate the role of Set7 in physiological and pathological processes. And this is not limited to metabolic disorders. And in fact, there have been a lot of studies that have examined the role of Set7 in cancer as well as inflammation. So while small molecule inhibitors like PFI2 may hold potential for therapeutic development, the process of translating that potential will depend on preclinical research findings like those published by our group in JASN, and that will depend on additional preclinical testing to evaluate safety, efficacy, and pharmacokinetics.
Well, my last question, I think you partly answered, but maybe we could drill down a little bit more. So the mice treated with streptozotocin developed pretty rip-roaring diabetes with very elevated glucose concentrations. And these days, most patients, achieve reasonable control in many or most cases of their diabetes and don't run around with glucose of 500 all the time, although certainly some patients do. But it sounds like you think that the Set7 pathway is triggered as much by episodic elevations of glucose leading to persistent epigenetic modifications, so that the findings that you have would be applicable even if people weren't severely hyperglycemic continuously. Is that fair?
Yeah, I think that's a very nice summary. If albuminuria and glomerular structure are to go by, and they were dramatically improved in the diabetic Set7 knockout animals, then my initial feeling is yes. So let me put that into context. We know that albuminuria is a manifestation of glomerular filtration barrier destruction. But the molecular mechanisms that drive this process in diabetic kidney injury remain poorly defined, at least with respect to epigenetic pathways. And our studies have shown Set7 is expressed in podocyte and glomerular endothelial cell types that are functionally important in renal filtration. And the single-cell RNA-seq data allowed us to unravel the endothelial cell populations that were altered in the diabetic Set7 knockout kidney. And nowhere is this more evident than its impact on pathways such as extracellular matrix organization, as well as interleukins and TGF-beta receptor signaling observed in mouse and human glomerular endothelial cells. And in fact, previous studies have shown a context-dependent role for vascular Set7 in pro-inflammatory gene activation. And our studies suggest in diabetic kidney injury are also associated with hyperglycemia regulated by endothelial inflammation and progressive damage of the filtration barrier. Now, with respect to your question speaking to the benefit of targeting Set7 in the progression of diabetic kidney disease, my feeling is the Diabetes Control and Complications Trial, or the DCCT, and its follow-up study, the Epidemiology of Diabetes Interventions and Complications Trial, referred to as the EDIC, these landmark clinical studies investigated the long-term effects of intensive glucose control on the development and progressions of complications in type 1 diabetes. Now, while these trials primarily focused on the effects of glucose control interventions, including the development of hypoglycemic memory, the role of specific epigenetic regulators such as Set7 was not directly tested. However, the findings from the DCCT and the EDIC trials have provided insights into the mechanisms underlying the development and progression of diabetic complications, and that includes diabetic kidney disease. And these studies have contributed to our understanding of the importance of long-term glycemic control and the concept of metabolic memory, where the effects of prior glycemic exposure continue to influence diabetic complications even after glucose levels are normalized. Now, given Set7's regulatory control in hypoglycemic memory and its potential role in diabetic kidney disease development, it's conceivable that Set7, along with other regulatory determinants, may play a role in the molecular mechanisms underlying hypoglycemic memory. In any case, further research exploring the role of Set7 in development of diabetic complications, including DKD, may help elucidate the mechanisms underlying metabolic memory and identify potential therapeutic targets for treating or preventing diabetic complications.
You know, one thing that just occurs to me that the other side of this might be explored in the Kidney Precision Medicine Project. I'm sort of peripherally involved in that because I'm on the Observational Study Monitoring Board. But one of the groups that they're biopsying is people with long-term diabetes who don't develop kidney disease, and the interest is why. And it'd be interesting if they had different regulation of Set7 that protected them, perhaps less elevation that protected them from the development of DKD over the long term. So lots of interesting things to pursue here. And again, I want to thank you so much for joining us today. We're really excited to have your paper in JASN. We're especially excited to have papers from throughout the world, where we really want JASN to continue to be an international journal, and it's great to have your paper as part of that. And we look forward to contributions in the future.
Many thanks, David. I really enjoyed the review process at JASN, the feedback, and the podcast.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.